Hello, dementia interested people. Today I'd like to talk about a very special component of all of the cells in your body, a special functional unit that turns out to be very important for dementia, particularly the dementia of children. I think I can best introduce the topic by posing a couple of questions. That is, do our brain cells have stomachs? And if they did have some stomachs, can they get a serious case of indigestion? Well, as you might imagine, the answer I'm looking for is yes. Yes, they do. The stomach of cells is called the lysosome, which actually is a scientific word derived from Greek. Two little parts here, lyso, meaning to loosen or break apart, and some meaning body or object in biological terms. So this is something within cells, a body within cells, that can break apart molecules, just like your stomach begins the process of breaking apart your food when you're eating. Now, I've drawn a very simplified diagram here to illustrate the idea of the lysosome. This is a crude diagram of part of a cell. Here on the upper part, you can see this line which is meant to represent the surface of the cell, the outer surface of a cell. And as you can see, parts of that outer surface can become pinched off and form small little globules. We would call these vacuoles in biological terms. So they're basically little spheres that are bounded by lipid membranes, or little hollow fat molecule, hollow fat globules, if you will. Anyway, so things outside the cell can actually be taken into the cell in this way, and eventually they're delivered to a particular compartment, a particular organelle, as we would refer to it, a part of the cell called the lysosome, and here it is, receiving molecules from outside of the cell. The lysosome can receive nutrient molecules from outside of the cell, but it's also very important for recycling the components of cells, because cells are made up of molecules, and a cell is constantly having to deal with the need to change itself. Either it needs to repair the breakdown of the molecules which make it up, or it needs to behave or change its structure in some way in order to cope with changes in the environment or your other body's needs that might need it to change its function or structure in some way. So cells are constantly needing to change, and that means that they need to break down the components that they've used to build up a structure and then reuse them. I mean, otherwise it would be very inefficient if they could only break things down and then take in new nutrients from outside. So cells are expert at recycling themselves, and they actually have a word for this. We call it autophagy, or other people call it autophagy, but I prefer autophagy. And autophagy literally means self-eating, the ability to eat yourself. And that's what a cell can do. It can enclose parts of the cell, any part of the cell almost, in something called an autophagic vacuole. And then that autophagic vacuole can be merged with the lysosome, and the contents of that autophagic vacuole will then be broken down in the lysosome and recycled and used again. So since the lysosome is involved in bringing in large molecules from outside the cell and breaking them down into small components to then be used, or it's also involved in taking large components from the cell itself and breaking them down into small components so that they can then be reused. Since it's involved, in other words, in central provision of nutrients to the cell, central provision of building blocks, it makes sense that the lysosome actually is involved in the monitoring of the nutrient status of a cell and in the regulation of processes that affect the nutrient status. Now, interestingly, the lysosome needs to be quite acidic in order to function properly, just like our stomachs need to be quite acidic. It needs to have, in chemical terms, a pH of 4.5 to 5. pH is the way we measure how acid or basic something is. Now, this acidic environment in the lysosome is created by a very large molecular machine, a kind of pump called the vacuolar ATPase. And what the vacuolar ATPase does is it uses a fuel called ATP, and it breaks that down into ADP. And using the breakdown of that fuel, it actually takes hydrogen ions, protons as they are, they're basically hydrogen atoms where the electron has been removed, so it's just a nucleus, and it pumps it into the lysosome and builds up the concentration of these protons. And that creates the acidic environment of the lysosome. The lysosome also contains a large number of, we can think of them as additional molecular machines called enzymes. Enzymes are biological catalysts that guide biochemical reactions. In other words, in this case, the breakdown of particular molecules into smaller molecules. Now, interestingly, these lysosomal enzymes, they need to function at low pH. They've evolved to function optimally at low pH, which is the environment within the lysosome. And problems occur if the acidic environment of the lysosome cannot be maintained, because that means that the enzymes that are working within the lysosome can't work as efficiently as they normally would. If one of the enzymes in the lysosome does not function properly, for example, maybe due to the fact that the gene coding for that enzyme is mutated, then the molecule that the enzyme would normally break down can accumulate in the lysosome. This accumulation of a molecule that cannot be broken down in the lysosome can have very deleterious effects on lysosomal function. The type of cell affected by this problem with lysosomal function will depend on the particular molecule that cannot be broken down. So, for example, the muscle cells of your body, and liver cells as well, like to store energy in the form of a molecule known as glycogen. It's a kind of large animal carbohydrate molecule. Now, there's a particular genetic disease where a person lacks the ability to break down glycogen in their cells, and the disease is mainly seen as affecting muscle tissues, since those are the cells that show the greatest accumulation of glycogen, since glycogen is such an important molecule in muscle cells. Another disease caused by accumulation of a molecule that cannot be broken down in the lysosome is Sanfilippo syndrome childhood dementia. Now, this is caused by accumulation of a molecule known as heparan sulfate. Interestingly, heparan sulfate is mainly found outside of cells. Of course, these molecules are produced within cells, but are then placed outside of cells, and then on an ongoing basis, they need to be turned over and replaced, just during the normal maintenance of a cell. Heparan sulfate is a large carbohydrate molecule. In that way, it's similar to glycogen. But in the case of heparan sulfate, it's actually attached to a protein backbone to form an even greater molecular structure. Now, if cells are unable to break down heparan sulfate, it will accumulate in lysosomes. And this particularly happens in cells of the brain, because some of the cells of the brain have very large surface areas, and hence they have very large volumes of extracellular matrix that need to be maintained. I'm thinking particularly of the cells that make the myelin sheaths that are wrapped around axons and help information transmission in the brain. These cells are known as oligodendrocytes. Collectively, these diseases that occur when a molecule cannot be broken down in the lysosome and so begins to accumulate there are called lysosomal storage disorders. Now, they're not actually an inability to store something, they are an inability to break something down. So the name can be a little confusing. Sanfilippo syndrome childhood dementia is a lysosomal storage disorder where the accumulating molecule involved is heparan sulfate. Tragically, it causes destruction of the brains of young children. Now, one of the research questions that we're involved in my laboratory is actually looking at whether the mechanism that causes the destruction of the young children's brains is similar to the mechanism that's giving problems in inherited early onset Alzheimer's disease. But that's something that we'll talk about for sure in later videos. And now for a distraction. While just checking up some of the facts for this particular video, where I wanted to draw parallels between your stomach and the action of lysosomes in cells, I found out something rather surprising, and I thought I'd just tell you that as well. It's a bit of a divergence, but that's what makes science interesting. What I was wondering was if the pH of the lysosome, when it's functioning properly, is around 4.5 to 5, how does that compare with the human stomach? How acidic actually is the human stomach? So I did a bit of searching in the literature and eventually was taken back to this particular paper from 1990 in the journal Pharmaceutical Research by Dressman and colleagues. And you can see stated here, and I'll just show the abstract, it says, in the fasted state, the median gastric pH is 1.7. Now this is remarkably low. It's a very acidic environment. Furthermore, I found another paper by Fujimori in the World Journal of Gastroenterology from 2020 that summarizes some work by others looking at the pH of the stomachs of various animals, and that showed something quite remarkable. And that is that the human stomach pH is actually very acidic compared to most other animals. So on this particular diagram, which is figure 1 from Fujimori's paper, we can see the information summarized here. We have herbivores eating plants, of course, and looking at 27 different ones of those. And the average stomach pH there was around 5. For carnivores, it's lower, where they're digesting meat, and it's around 3. And the same applies to omnivores. They're eating both meat and vegetable matter. And then there's the group of scavengers, the carrion eaters, that are eating, for example, dead animals, and which typically have very acidic stomachs because they need to kill the bacteria that are growing in these dead animals. And actually humans have a gastric pH which is comparable with the carrion eaters. And this leads the author to speculate that maybe in humans evolutionary history, we may have been carrion eaters ourselves and so required this low pH. So I thought that was rather remarkable. Anyway, the main point of this video was to tell you about the lysosome. And now that we know a little about it, we can begin to talk about childhood dementia, in addition to our videos on Alzheimer's disease. And so I'll see you at the next one, hopefully. Goodbye.